Hi everyone, my name is Imran Razik. I'm a PhD student at Ohio State University, and today I'll be presenting some of my dissertation research, specifically looking at the relationship between forced proximity and the choice of cooperation partners in vampire bats. So starting off, spatial assortment or spatial proximity is tightly linked with cooperation on both developmental and evolutionary timescales across a wide variety of taxa, including microbes, humans, and many other animals in between. However, it's important to note that proximity can be both a cause and a consequence of cooperation. For example, proximity might lead to repeated interactions, which in turn might lead to frequent cooperation. So say you have a focal individual in a two-dimensional landscape, the slide here. This individual may at any moment in time be surrounded by conspecifics at varying distances. However, it's perhaps likely that it would most frequently interact and cooperate with those that are simply nearby, just like this. Now, on the other hand, you can also have a causal relationship in the reverse direction, where cooperation in the past can maintain repeated interactions and close proximity between partners. So in this situation, if our focal individual already has preferred cooperation partners, which are further away, say these two over here, our focal individual might actually move towards or associate with these partners more through time. Now, if we just focus primarily on the first hypothesis here, where proximity might lead to cooperation, we know that testing for this experimentally can be difficult, especially when using correlational data sets or working with subjects that already have well-established social relationships, such as many living primates in the wild. To actually test this hypothesis experimentally, there's two main things that need to be done though. First, you need to, of course, manipulate proximity between random subjects, preferably non-kin strangers. And second, you need to describe long-term changes in the cooperative behavior of these subjects after the manipulation ends. So the keyword here being after. And one illustrative example of this concept actually comes from research on college students, which found that randomly assigned first-year college roommates actually showed a higher likelihood of being friends after graduating. So in other words, proximity induced by short-term spatial constraints might promote the longer-term maintenance of a social relationship, even when spatial constraints change or proximity is no longer forced. Now, this hypothesis hasn't really been rigorously tested in non-human animals. However, a species well suited to testing this hypothesis is the common vampire bat. Vampire bats are known to form relationships where they allogroom, share food, and associate with one another through time. And as it stands right now, there are a few studies that suggest vampire bat cooperation could be driven, at least in part, by proximity and familiarity bias. Jerry Wilkinson was the first to describe how repeated coercing associations could predict food sharing rates in wild colonies. In captivity, bats that were introduced for the first time in small cages were faster to develop new food sharing relationships than bats first introduced in a larger cage. And also, bats that were housed together in captivity for 22 months were found to preferentially associate with one another after being released back into the wild. So, of course, all informative studies here, but they actually failed to provide conclusive evidence in support of our focal hypothesis because they lacked either proper controls, sample sizes, or the means to track longer-term effects, at least greater than 9 days. As a result, we're still left with this really important question of, will short-term forced proximity induce long-term allogrooming? And this is the question we set out to answer with our study. So what do we do here? Well, we first captured wild vampire bats across 3 different sites in Panama, so marked here, such that bats between sites were unrelated strangers. After catching these bats, we then released them into an outdoor flight cage at the Gamboa facility of the Smithsonian Tropical Research Institute. We then used 3 infrared surveillance cameras within this outdoor flight cage to sample their allogrooming interactions for 6 weeks to essentially get their quote-unquote pre-treatment allogrooming network, which you can see here. Bats from different sites have nodes that differ in both shape and color, and the wooded gray lines or edges between nodes represent the mean allogrooming rates for each dyad. At the end of this first 6 weeks, we then separated the bats into 7 different triads, so each consisting of 3 bats from different sites, and these triads were housed within smaller plastic observation cages for just 1 week. At the end of this treatment phase, we then released all the bats back into the main flight cage and continued to sample their allogrooming interactions for an additional 9 weeks, in total sampling the behavior of these bats for 682 sampling periods across 114 days, so that's about 6 hours per day. Now, to actually test our hypothesis and effective forced proximity on allogrooming rates, we compared changes in mean grooming rates from pre-treatment to post-treatment for dyads of 3 different types. These include our 21 test dyads, which are new pairs forced into proximity, 126 control dyads, so new pairs not forced into proximity, and 63 familiar dyads, which consist of bats from the same site. Now, what I'd like to do is kind of show you our general or overall prediction for how these different dyads would respond to the treatment phase, and then I'll lead right into the results afterwards. So if you direct your attention to this plot, On the x-axis, we have our different dyad types, familiar, control, and test. On the y-axis, we have the change in mean allogrooming rate from pre-treatment to post-treatment, as I mentioned. At first, for familiar dyads, we didn't expect any major change in grooming because we assumed these would mostly be pre-existing relationships, less likely to show significant change over the course of the study. Second, we predicted that the mean grooming rate for control dyads would likely increase, simply owing to the natural development of new relationships over time. And finally, we predicted that we would observe the greatest overall increase for our test dyads because these are, of course, those that are forced into proximity within those small cages. So again, this is the overall pattern for what we predicted. Now, what did we find? Well, we found some pretty strong support for our prediction. Mean allogrooming rates increased 63% more among test dyads relative to control dyads, and this is, of course, visualized here with the actual data now. We have our mean grooming rates with these black points with corresponding bootstrapped 95% confidence intervals for each dyad type. So really exciting result here, but what do we make of it now, and kind of like what questions remain going forward? Well, first of all, our result does seem to bolster the idea that cooperation and social preference can be driven by proximity and familiarity bias. And if we look to some other relevant examples in the literature, we know that early life experiences in many birds are known to provide the foundation for kin discrimination and social preferences much later in life. It's also been found that newborn bats raised in separate groups will preferentially associate with and groom fellow groommates, even those of completely different species, after being released into a common flight cage, kind of similar to what we did here. However, as I mentioned, these are cases pertaining to early life experiences and early life familiarity. Less understood is how proximity and familiarity might play a role in social bonding among adults. Even further, how long does proximity need to be in order to create a lasting effect on the pairwise relationship? In our study, we observed an effect after only 7 days, which really isn't very long, and even further, the effect was still apparent at the end of our experiment, 65 days later. Nevertheless, although these are, of course, important questions, what I'd actually like to end this presentation with is another really interesting hypothesis that social bonding may depend on how individuals perceive their shared experiences with others. For example, if we consider the possibility that bats in our experiment perceive the treatment phase as stressful or negative, this very well could have facilitated or reinforced partner choice between cage mates, even when spatial assortment changed and they were no longer as limited to who they could interact with. If so, this would by no means be the first evidence to suggest support for this hypothesis. In fact, one great example comes from research on guppies, which found that individuals exposed to an environment where they perceived higher predation risk exhibited stronger partner preferences for one another than those that were not. So overall, definitely excited to see where future research on this topic will take us. And if anyone is looking to answer some of the same questions that I covered here, if you know of any similar studies, I'd love to hear from you. And with that, I'd like to quickly thank my co-authors listed on this presentation: Bridget Brown, my lab mate; Rachel Page, my Stri Fellowship advisor; and Jerry Carter, my PhD advisor, as well as all of Team Vampire who helped out in 2019, the Gimbo Bat Lab, and my funders listed here. Thanks for watching, and I'm happy to answer any questions.